We're going to run through the male reproductive system PowerPoint. If you've got your handout ready, this shouldn't take too long. We start by looking at some of the anatomy, and then we're going to get into some of the physiology about actually producing gametes. It's interesting when we get to the reproductive system because this is the only organ system that isn't responsible in maintaining homeostasis. So all of the other ones, like we were just talking about the urinary system, its function with the respiratory system is to control things like hydrogen ions of the bloodstream. None of that happens here with the reproductive system. The goal is just to produce gametes. We're going to separate primary sex organs from secondary sex organs. The primary sex organs in males and females are going to be the site of gamete production. So this is going to be the testes in males and the ovaries in females. Secondary sex organs aren't going to produce gametes, but they're going to house or store or transport these gametes along their journey. So accessory sex organs or secondary sex organs do the latter, like we said, transport, store, maintain these gametes along their journey. In males, primary sex organ is the testes. Secondary sex organs are going to be a list of glands. We're going to see things like seminal glands, prostate gland, and bulbo urethra gland. I believe all of those are on the lab list and in the class handout. So let's look more closely at some of these structures, and just let me remind myself of where we are on the slides. There's a model in lab that looks something like this, and we had seen it before. The structures that we had seen before on the male reproductive model that looked like this, if I remember correctly, we saw the testes, where the site of sperm production. We're going to look inside there to see exactly where sperm production takes place. Then kind of bordering the testes, there is this epididymis. We're going to see that sperm, after they're produced in the testes, are going to hang out in this epididymis where they mature and may be called upon, and if so, muscular contractions of this epididymis are going to squeeze these sperm through this tube that we previously called the vas deferens or ductus deferens, and this is going to make its way in through this spermatochore, kind of into the abdominal cavity. And this is where I really wish I could grab one of those male reproductive models. We saw this before. You can see the ureter actually travels across the top of the urinary bladder and then around the back side of the urinary bladder. For a second, it crosses right next to this little tube. That's the ureter. That is coming from the kidneys and filling up this urinary bladder. So we've got the urinary bladder, and then you've got this vas deferens that goes right on top of the urinary bladder and around the back side. In males, this vas deferens around the back side of the urinary bladder, this vas deferens is going to merge with the first accessory gland that we're going to see. I know nothing is labeled here, but this is the seminal vesicle, or sometimes called the seminal gland. It sits around the back side of the urinary bladder, and it's going to contribute about 60% to the volume of semen. Sperm are produced here in the testes, but semen, the stuff that is transferred during copulation, is a mixture of sperm and nutritious fluid, and then some of it's protective fluid, some of it's lubrication fluid. So we'll talk about each of those accessory glands and what they produce. The seminal vesicle produces about 60% of the volume of semen. I'm giving it away here. It's a sugary rich substance that's going to nourish these sperm along their journey. The next gland that we can see here is the prostate gland. It's this walnut-shaped gland. It sits underneath the urinary bladder. It's important for a couple of reasons. One, we're going to see that it produces an alkaline substance that is going to try and help. It accompanies the sperm along their journey, and if they're being transferred into the vaginal canal, they're going to encounter an environment that is acidic, and the sperm are not going to like that. So to help protect them from this acidic environment, we'll see secretions from the prostate are alkaline to try and neutralize that pH of the environment that they might be going into. So that's the prostate gland, and then the last gland we can see down here is the bulbo urethra gland. There's not a model in lab that shows this very well, in my opinion, but the slides here show us a decent job of this bulbo urethra gland. It sits at the base of the penis. Its job is to produce a lubricating mucus-like substance that cleanses the urethra and lubricates the urethra prior to ejaculation. We can see the copulatory organ, the penis, there in this cross section. They're trying to show the erectile tissues in two different ways. One of them, this dorsal patch of erectile tissue, they've almost kind of put this striped-like appearance through it. We're going to call that the Corpora Cavernosa. I'm jumping ahead here. Then the corpus spongiosum, the lower level of erectile tissue in the penis, the copulatory organ, has the urethra that runs through it. So it's sometimes called the spongy urethra. So let's see, maybe from another view. This view shows us from an anterior view the same thing that we were looking at. The testes, we can see a little bit of that epididymis. What this slide shows relatively well is that things like we can see the vas deferens here, but accompanying the vas deferens, there are going to be blood vessels, there are going to be nerves, and they're all wrapped together in this structure called the spermatochord. Here's our spermatochord. And together, this whole little cable that's connecting nerves, blood vessels, vas deferens, it all enters this abdominal cavity through this inguinal canal. So spermatochord goes through the inguinal canal. This is an example of a herniated, or what happens in somebody that has a hernia. There's a piece of the intestines that has squeezed through this inguinal canal. What else can we see? There's a couple of things to point out. This is coming from the lecture handout. There is the fleshy pouch that the testes are housed in. We're going to call that the scrotum. But there's going to be two muscles that we need to be aware of. And there's two muscle layers that I'm trying to point out. The one that is closest to the testes is going to be called this cremaster muscle. This cremaster muscle, it's on the lecture handout. What we need to know about the cremaster muscle is these are bands of skeletal muscle. What's interesting about the skeletal muscle, remember that we are in voluntary control of the skeletal muscle. So the cremaster muscle, males can lift, let me say that, I'm going to have to erase this part of it. But there's two things that happen, I guess, in the testes. This whole business of sperm production is very temperature sensitive. So the two muscles that I'm pointing out here are going to have the dual role of either elevating the testes closer to the body, warming them up, or dropping them further away from the abdominal wall to cool them off. The muscle that lifts the testes, this is the cremaster muscle. Again, it's a skeletal muscle. You're in voluntary control of it. Contracting this muscle lifts the testes. Relaxing this muscle is going to drop them further away from the abdominal wall. Then if we look in the walls of the scrotum, so there's the outer layer there is when we're looking at the layers of the scrotum. The outer layer is the skin, but there, just underneath the layer of the skin is this muscle that's a thin layer of smooth muscle called the dartus muscle. Being smooth muscle, you're not in voluntary control of this. Its job is to, again, relax and allow these testes to drop further away from the abdominal wall, or it can contract and push those testes up closer to the abdominal wall. And males that have ever gotten into extremely cold water will experience this dartus muscle contract and try and elevate those testes closer to the abdominal wall. The other thing that we can see from this picture, and I think it's on the lecture handout, is this Popiniform Plexus. This Popiniform Plexus, that's a funny word, but what it describes is a network of veins. It's almost like a net of veins, and they wrap around the artery. You can't see the artery very well, but if we think about this, we know that the artery is delivering fresh, oxygenated blood towards the testes. And this blood is going to be relatively warm because it's coming from the core of the body. And we can see that surrounding this artery that's delivering warm blood to the testes, that artery is surrounded by a network of veins, this Popiniform Plexus. The job of that network of veins is to pull heat off of the warm blood that's going towards the testes. What else do we need to say about anything in there? Maybe this is one of the only other slides that actually shows the prepuce. This is the foreskin that covers the glands, the head of the penis. So we can see the shaft of the penis, we can see the head of the penis. There is the foreskin in uncircumcised males. We would refer to that as the prepuce. I think the other, so again, prepuce is still present there and still present in this one. I think it's not easy to see on the models because I think they kind of have it pulled back or it's not clear. So prepuce is the foreskin around the glans penis. Let me know if I'm leaving anything out. Let's look at that erectile tissue real quick because we're going to see that there is corresponding patches of erectile tissue in the female reproductive system, just like we see it in the male reproductive system. And that's also kind of interesting to think, and I don't have these pictures here, maybe I'll bring them in next time, that show that developing embryos start out with the same series of tubes. And depending on whether or not those tubes are bathed in testosterone or whether or not those tubes are bathed in estrogen, they develop in different ways. So looking at male structures, we can see, first off, if we break this copulatory organ down into 3 segments. There is the root of the penis, which kind of like the root of a plant is underneath the ground. You can't see the root, so the root of the penis is inside the body wall. It's going to include the bulb of the penis. That's where those bulbo-urethra glands deposit their secretions into the bulb of the penis. Then we can see the crura of the penis. These are the little flanges that stick out on either side of the bulb of the penis. Then when we get into the shaft of the penis, this is obviously the part that sticks away from the body wall. Then the expanded tip is what we're going to call the glans penis. This one you can see still has the foreskin around it. But where we're going with this, inside this copulatory organ, we should be able to distinguish the 2 patches of erectile tissue. One type of erectile tissue is called the Corpora Cavernosa. Notice the ending there, corpora, that's plural. There are 2 of these corpus cavernosa. So they collectively often refer to these as just the Corpora Cavernosa, plural. There's 2 of them. They sit on top. They're the dorsal patches of erectile tissue. And then underneath that is one single patch of erectile tissue called the corpus spongiosum. The corpus spongiosum is what has the urethra traveling right through it. You can also see the glans penis is also made from this corpus spongiosum. Here's a side view of that, and for whatever reason that looks like a face with 2 big eyeballs to me, but that's not what this is trying to show. If you can't unsee that face with eyeballs, then you'll notice these 2 big, what look like eyeball structures. Right in the center, that is an artery. And there is a bunch of this spongy tissue that, like a balloon, can get inflated with blood. And that's all that Viagra does, is it dilates these blood vessels and allows these patches of erectile tissue to inflate. The Corpora Cavernosa, this is one corpus cavernosa. There's the other Corpus Cavernosa. Together we refer to them as the Corpora Cavernosa. And then underneath that we can see what looks like the mouth part is the Corpus Spongiosum. And that mouth is actually the urethra that travels through the Corpus Spongiosum. We could also include, like we said, the glans penis is also made up of this Corpus Spongiosum erectile tissue. Again, showing the same thing, we can see those patches of erectile tissue, corpus spongiosum with the urethra that travels all the way through it. And then we can see, kind of cut away, there are those 2 patches of the corpora cavernosa on top. What I like about this slide is that it shows those accessory structures, the seminal gland or seminal vesicle. We can see the prostate gland and then the bulbo-urethra gland. We mentioned before, seminal vesicle is going to contribute about 60% towards the volume of semen. It's mostly a sugary rich substance that's going to be a nutritious fluid for the sperm. So they get energy along their journey. Then after sperm enters this prostate gland, again, they're pumped into that prostate gland with this sugary rich fluid from the seminal vesicle. So they've got food. While they're in this prostate gland, they get the protection from the prostate secretions. It's mostly an alkaline fluid, helps protect them into the acidic environment that they may go into. And then the last gland, that bulbo urethra gland, we said cleanses the urethra and lubricates that urethra prior to ejaculation. Here's the prostate gland. Not that we have to know the anatomy of it, but you can see all of these extensive branches that allow those secretions away into the urethra. This is the slide that I wanted to see. What is important about the prostate is it also has to direct traffic. There's a dual purpose for the urethra in males, and that is not only does it have to deliver sperm during copulation, but the urethra also has to drain the urinary bladder. So this dual purpose, kind of what it's being used for, is determined by the prostate. So when we look in the prostate, there's kind of this little Y-shaped structure, and that little bit, that duct through the prostate, we're going to call the ejaculatory duct. It's just that little part right there. So this ejaculatory duct is going to deliver semen into the urethra. The urethra, we mentioned last time, is a lot longer in males. It drains this urinary bladder. And on the lecture list, there's actually 3 parts to the male urethra that we need to be aware of. There's the first part of the urethra that travels through the prostate. It's called the prostatic urethra. You see it on that the lecture handout. Then after we go through the prostatic urethra, it's kind of faint on this one, but there's a little muscle. It's a thin layer of muscle that the urethra has to travel through to get out of the abdominal wall. So through the prostate, it's the prostatic urethra, but then just right there, right as it crosses through this layer of muscle, that's what we call the membranous urethra, very short. And then once we're through that body wall into the spongy cavernosa, it's either called the spongy urethra or penile urethra. And that takes us all the way out to the urethral orifice. I think there is a question in the lecture handout where you have to trace the pathway that sperm would take from their site of production, from the testes, through the epididymis, through the vas deferens, through this ejaculatory duct, and then through the membranous urethra, through the penile urethra, and eventually out the urethral orifice, which is where we are going in the next slide. Are we doing all right so far? Trying to catch back up. We don't have very many more slides to go through, but we're zooming in to the primary reproductive organ, the site of sperm production, and that's going to be inside the testes. This is the second page of the lecture handout. There's a little flow chart that lists 13 things that we should be able to organize in the correct order from the site of sperm production to where they leave. There's actually a nice picture of that spermatochord where you can see that Popiniform Plexus, that branching network of veins that surround the artery that's delivering warm blood down to the testes. So let's jump in here. The next slide shows that this is a cross-section through one of the seminiferous tubules. Inside that seminiferous tubule, if you think of this tube as like a garden hose, and we just chop the garden hose in half, and we look inside the hollow tube, we can see that this garden hose is lined in its perimeter by a layer of cells that are dividing rapidly into developing sperm. So we're going to look, in a second we'll probably draw on the board a cross-section through this garden hose and list those cells that are dividing. But I'm going to back out of that just for a second, back to this picture. What we were just looking at is a cross-section through one of those coiled tubes, these seminiferous tubules. Notice how each of those seminiferous tubules, this is not an open-ended tube. Each seminiferous tubule is kind of a closed tube that ends up opening to, it's very short. It's a short little structure that connects these seminiferous tubules to this orange structure. The orange structure is called the rete testes. This rete testes is going to receive newly produced sperm from each of these lobes of seminiferous tubules. So there's a bunch of these lobules of seminiferous tubules. Each one is producing lots of sperm. The sperm, as it leaves this seminiferous tubule, is going to go through this tiny little tube called a straight tubule. You can see how each of those get their name. It's just a straight little tube that connects the seminiferous tubule to the rete testes. So if you're sperm, you start off in the seminiferous tubule, you travel through the, let's try this again. You start in the seminiferous tubules, you travel through the straight tubule as you get out of these seminiferous tubules, and now you're into this rete testes. The rete testes, you can see, are receiving sperm from all of the seminiferous tubules. And sperm, as it travels through the rete testes, is going to make its way into, it's getting towards this epididymis. But in order to get to the epididymis, it has to travel through these other little tubes that we call an efferent ductule. Efferent is away, so these are taking sperm away from the rete testes and into the epididymis. These efferent ductules are going to lead to what we call the head of the epididymis. So the head of the epididymis is going to contain the most immature spermatids. As these spermatids make their way from the head of the epididymis down towards the body of the epididymis, and then eventually down here to the tail of the epididymis, the whole time they're moving through this epididymis, they are maturing into mature sperm. And mature sperm are going to hang out down here in the tail of the epididymis and may or may not be called upon. If so, like we said, muscular contractions of the epididymis are going to squeeze them up through this vas deferens and into the abdominal cavity. Epididymis all the way down to the tail of the epididymis, and then when we start to make our way back up, we refer to that as the vas deferens or ductus deferens. So from the flow chart, we can get halfway. This is number 22. It starts there in the seminiferous tubules, then it goes to the straight tubule. From the straight tubule, we enter the rete testes. From the rete testes, we enter the efferent ductule. From the efferent ductule, we go to the head of the epididymis, the body of the epididymis, and then the tail of the epididymis, which then turns and leads into this vas deferens or ductus deferens. To see the rest of the story, we need to jump back to this picture. This vas deferens, we know, goes around the back side of the urinary bladder where it merges with this seminal vesicle. If we follow the sperm, though, the sperm are going to go through this vas deferens and they're going to enter this ejaculatory duct. It's right there in the ejaculatory duct that they get fed by secretions of those seminal vesicles. So sperm, once they go through this ejaculatory duct, they're now in this part of the urethra that's shared with the prostate. So we call it the prostatic urethra. Then this urethra goes through that membrane. So the sperm travel through the ejaculatory duct, through the prostatic urethra, through that membranous urethra, and are now in this spongy urethra or penile urethra. I think it calls it spongy urethra in your lecture handout. And then from the spongy urethra, we can see that goes all the way out to the urethral orifice. And most of the other details we can draw on the board. I think that takes us through some of the male anatomy stuff. So back to this picture, we know that sperm production takes place inside those seminiferous tubules. If we were to look at a cross-section through that, it is a hollow tube that we can see is lined in the perimeter by a bunch of these cells that are dividing into what are going to be mature sperm. This just reminds us of the flowchart question that we just went through. So hopefully we check that off the list. And the last few slides that we have are to actually look at a cross-section of the seminiferous tubules. This slide that we're seeing, that's a cross-section through one of those tubes, one of these seminiferous tubules. Around the perimeter, you can see cells that some of those are dividing into what are going to be mature sperm. And then there are two other cell types that we need to be aware of. And before I even go to the next slide, let's see if we can draw this. So these are all cells that you see lining the seminiferous tubule. So to get this diagram going, I'll start with this color. And I'll just let this represent the seminiferous tubule. This seminiferous tubule we're going to see is lined by a couple of different types of cells. I'll start by drawing these cells first. I'll put another one over here just for fun, and we're going to label this thing a spermatogonia. This spermatogonia is going to be one of the cells. In fact, when we start talking about hormones, we're going to see that follicle stimulating hormone, that's one that we talked about before. Follicle stimulating hormone is going to work on these spermatogonia. It's going to stimulate these spermatogonia to divide. So mitosis is stimulated in spermatogonia by the follicle stimulating cell. What it's going to do, in fact, let's just draw that over here. Let's put some of the follicle stimulating hormone affecting this spermatogonia. These spermatogonia are going to divide. Whenever they divide through this process of mitosis, they always replace themselves. So they make a copy that stays along the edge of this spermatogonia. And then the other cell, this other diploid cell, is going to start to make its way towards the middle of the seminiferous tubule. This second cell is still called a spermatogonia. These spermatogonia are going to continue to divide. Again, as long as there's follicle stimulating hormone, that's going to cause these spermatogonia to continue to divide. Mitosis gives us identical copies of cells. So maybe let me come over here and remind us of something, because it's maybe not something we've talked about a whole lot, but that lowercase n just represents sets of chromosomes. So haploid cells just equal one set of chromosomes. And our diploid cells, we call these diploid, and they've got two sets of chromosomes. I only mention that because our normal body cells are these diploid cells. We'll give it a little green check mark. Normal body cells. And that is relevant because we had just gotten through in Unit 3 talking about the immune system. One of the jobs of the immune system is to do what's called immunosurveillance, and that's to look at normal body cells and say, oh, that looks like a normal body cell, it's fine, or that doesn't look like a normal body cell, maybe we should take it out. We're going to see this whole business of making gametes is very temperature sensitive, but it's also tricky because you have to do this without the immune system seeing these cells that are not like normal body cells. Right now, spermatogonia are fine. They're diploid cells. They look like normal body cells. The immune system doesn't have a problem with this so far. But what's about to happen, these spermatogonia, as they divide, the next division of these spermatogonia, they're going to get given a different name. We're going to call these primary spermatocytes. Primary spermatocytes are still diploid cells, so the immune system says no problem with those. But primary spermatocytes are the cells that go through meiosis. Meiosis is a process of going from diploid cells down to haploid cells. So I'll draw each one of these cells going through meiosis. So that means this diploid cell is going to get split into haploid cells. And not that we have to know the details of meiosis, but for every one cell that goes through meiosis, we're going to see that it's going to produce 4 of these haploid cells. There's 2 stages to meiosis. There are 2 cell divisions. I haven't done a very clean job of drawing those, but all of these haploid cells, these are going to be what we call secondary spermatocytes. The key difference is that secondary spermatocytes are all haploid cells. The immune system recognizes that these are not like normal body cells and sees them as a target and wants to take them out. It's these secondary spermatocytes that if they don't get taken out, they're going to end up developing these tails and they're going to end up dropping into the lumen of this hollow tube. Maybe there's just enough space for me to draw this lumen, kind of the hollow center of this seminiferous tubule. And we can see the lining of this seminiferous tubule. There are several cell types that we need to be aware of. So far, we've only shown one cell type in the wall of this seminiferous tubule, and that's the cell that divides into developing sperm. We're going to call these immature sperm spermatids. So they drop into the lumen, and like we said, they make their way through the seminiferous tubules, to the straight tubule, to the rete testes, to the efferent ductule, and then through the epididymis, and that whole journey they're maturing. We'll see what happens as they mature and organize into what they're gonna, their final structure. But let's say real quick, there are a couple of other cell types we need to be aware of. There's a cell that helps protect them from the immune system. This cell is called a nurse cell. And we can't see it very well in this diagram, but these nurse cells are diploid cells. I'm going to draw the nucleus of one of these nurse cells. It goes by a couple of names. In your lecture handout, you'll notice it's called a sustentacular cell, or nurse cell. I think some textbooks even call this a Serratoli cell. So sustentacular cell, Serratoli cell, or nurse cell maybe is a better name. What these cells do, there are several of them along the walls of the seminiferous tubule. And what these cells do is they have this huge amount of cytoplasm that they actually spread out. And it acts as a cloak. It surrounds these dividing spermatogonia, and it kind of cloaks them from the immune system. This sustentacular cell over here has cytoplasm that it's fanning out, and that cytoplasm is cloaking these dividing spermatogonia. I wish I would have shown the two cytoplasms merging somewhere else, because there's actually a little layer of tight junctions that kind of seal together the cytoplasm of these two nurse cells. Maybe I can put one down here. And its cytoplasm is coming in here. There's a series of tight junctions that seal together the cytoplasm of these nurse cells. It forms what's called the blood-testes barrier. It's like tight junctions. So these nurse cells, or sustentacular cells, have a very important job. If you don't have functioning nurse cells, then these secondary spermatocytes are likely to be seen by the immune system and taken out before they ever had a chance to drop into the lumen. Okay, we're doing good. We got two cell types in here so far, so let's add the final cell type. And to see that, I'm going to continue this. I'm just going to close off this seminiferous tubule, and I'm going to draw another seminiferous tubule kind of coming in here. Maybe we can have a little bit of the lumen. I'll draw another seminiferous tubule kind of coming in here. We can only see a bit of it. In between these seminiferous tubules, there are going to be patches of another type of cell. I'll just draw them in a green color. I'll put a little nucleus so that we can see them. These cells that are not actually within the lumen of a seminiferous tubule at all, they're actually in between these seminiferous tubules. We're going to call these interstitial cells. These interstitial cells are the ones that you find in the testes. Again, they're in between these seminiferous tubules. These are the cells that respond to luteinizing hormone. I'll just maybe fit this somewhere in here. Luteinizing hormone, we mentioned, comes down from that anterior pituitary gland. In the case of males, luteinizing hormone is going to affect these interstitial cells. It's going to cause those interstitial cells to turn around and pump out testosterone. Just barely squeezing it in there. So testosterone, we know, comes from the testes. Specifically, it comes from interstitial cells, only when those interstitial cells are being hit with luteinizing hormone. So those are the three cell types we need to remember. We have seen hormones that we mentioned previously, like follicle-stimulating hormone and luteinizing hormone. Funny, we're going to mention the same two hormones when we draw female reproductive structures, and we're going to see follicle-stimulating hormone is going to stimulate follicles to grow. Luteinizing hormone in females is going to lead to ovulation or the release of a secondary oocyte. I'm using some terms there that we're going to see the strategy in females of making gametes. There's the same thing that female reproductive system has to worry about, and that is when these gametes turn to haploid cells, how are we going to get them away from the immune system? Males, again, do this with the help of the sustentacular cells that share their cytoplasm and effectively seal off these dividing cells from anything else, so the immune system can't see them. Females, on the other hand, I know I'm not drawing anything yet, but we're going to see on the surface of the ovary, as soon as haploid cells show up, they're kicked off the surface of the ovary and into the fallopian tube. And, I mean, if they stayed on the ovary, the immune system would get them. So they're pushed into the fallopian tube. They may or may not get fertilized, but at least the immune system isn't going to get them. And in males, that's not the case. We see the sustentacular cell. So let's look at some sketches. In fact, we'll start with this sketch and then I'll go backwards because this is kind of like what I was trying to draw. You've got the wall of the seminiferous tubule and all of these dividing cells, those are the spermatogonia. Those are the cells that divide into sperm. Those cells can only do so with the help of the sustentacular cells. And you can see this is a nucleus of one of those sustentacular cells. There's another nucleus. You can see the gold-colored cytoplasm that they're sharing, and it cloaks each of these dividing cells from the immune system. Those tight junctions right there are what I tried to draw that seals together the cytoplasm of these sustentacular cells. That forms our blood testes barrier. The only cell that you can't see here are those interstitial cells. I think here are those interstitial cells right there. So they're outside the wall of this seminiferous tubule. We need to know, not really how to recognize them, but we need to know that those are the ones that produce testosterone when hit with luteinizing hormone. Okay, back to this thing real quick. You can see those dividing spermatogonia. We can see the cytoplasm of the sustentacular cell. And the previous slide is showing what I took some time to write on the board. Again, along the side of that seminiferous tubule, those cells are spermatogonia. And as they divide, there's always one spermatogonia that replaces itself along the wall of the seminiferous tubule. And the second cell is going to start to move towards the middle of the seminiferous tubule. And those cells that move towards the middle of the seminiferous tubule are the ones that we call the primary spermatocytes. These are still diploid cells, so the immune system doesn't have a problem with them. But when these primary spermatocytes go through meiosis and turn into haploid cells, we now have to call them secondary spermatocytes. This is the whole tricky part. So secondary spermatocytes are going to go through 2 stages of division in meiosis. We need to just know that for every one cell that goes through meiosis, 4 haploid cells are produced. And after we go through the female reproductive system, we're going to compare a little bit of the difference in gamete production between males and females. In males, for every one cell that goes through meiosis, 4 sperm are produced. We're going to see they contribute really 23 chromosomes to the embryo. The egg, on the other hand, has to contribute 23 chromosomes, all of the cytoplasm, all of the organelles. So and I know I don't have a picture of this, but we're going to see in female gamete production, for every one cell that goes through meiosis, only 1 egg is produced, and the other 3 really don't get any cytoplasm, don't get any organelles. All of those resources are put into one of these haploid cells, so kind of all the resources are given to one of those cells in hopes that it has the best chance of making it. Anyway, we'll see where that takes place, and we'll name all of those diploid or haploid cells. Let's see. This again, Same thing that I listed on the board before. What I like about this slide is that it shows the chromosome number there. So we can see anytime that we are still talking about diploid cells that have 2 sets of chromosomes, the immune system doesn't have a problem. These are either spermatogonia or primary spermatocytes. Primary spermatocytes are those cells that go through meiosis. And so after they go through meiosis, every one cell that goes through meiosis, we can see 4 haploid cells are produced. It's the whole haploid stage that has to be cloaked from the immune system. Okay, if we look at one of these maturing spermatids, how it organizes itself, that's this picture right here. We're going to see that the organelles like the mitochondria are going to line up just behind the head. These are going to act kind of as an engine that propel this flagella that accelerated in one direction. If you look at the head of the sperm, all it is a head that contains 23 chromosomes, and there's a little cap, an enzyme called an acrosome. That acrosome, if it hits the surface of the egg, is going to be able to eat through the wall of the egg and drop those 23 chromosomes. I think there's something in the lecture handout that wants you to know the acrosome is found on the head of a sperm, and it's that digestive enzyme that allows it to eat its way through the egg. We've seen that slide before, so we know testes, this primary sex organ is where sperm are produced specifically in the walls of the seminiferous tubules. And we know how those sperm get out from the testes through the epididymis, through this vas deferens, through each of those segments, through the ejaculatory duct, through the prostatic urethra. Right there, that's easier to see that little muscle layer. So that's the membranous urethra. It's very short. And then we're into the spongy urethra. But I think that is all of the slides for male. We got done just in time to see these things once again on the models.